What up, botanists? So we're going to look at germination, which is it's the very start of plant growth, and that's where we're going to focus today. So we're not only going to look at germination, we're going to look at what a seed structure is, okay, and how do we impact, how can we affect the germination process? All right, so you need to remember this. Photosynthesis is very simply carbon dioxide and water plus sunlight energy in to give us sugar, glucose, and oxygen. And aerobic respiration is the opposite. Aerobic means with air, so it means with oxygen. So you can respire the other way as well. All right. So this is our basic seed structure. We've got several bits here. We've got the seed coat, which offers protection. We've got the endosperm, which is this stuff in the middle here. We have the cotyledon, which is essentially the root leaves. We'll see those in a second. And we've got the plumule, which is the entire shoot system. Okay, and we've got the radicle, which is completely rad. We like that one, and that's the root system. So we've got the plumule and the radicle. So let's have a look at a couple things. So what is a seed? Basically, it's the fertilized embryo of a plant. They're the unit of reproduction of a flowering plant and capable of developing into a whole plant. So one seed is where the whole plant comes from. And yeah, it's the reproductive unit. So the seed coat provides protection, as we said. The endosperm is actually quite interesting. It is the nutrition source for a seed. So the embryo, which is the whole thing, I suppose, but the embryo, which is we're looking at here, but the whole thing really counts as the embryo. But this stuff here is what turns into the plant. That needs a food source. As you can see, it can't photosynthesize yet. There's no sunlight getting in there. So it's the food source, the endosperm. That's this area here. The plumule is what turns into the shoot system, so that's everything above the ground for the plant. And the radicle is what's below the ground, and a cotyledon. These are the first leaves of a seedling plant. Now, we actually use these as a way to segregate and divide it. So it's used as a taxonomic structure. So we use it to define the plants, which we'll see in a second. So angiosperm, as we've discussed, is a flowering plant. So angiosperm is going to be divided into two categories, and as I said, we're going to use the cotyledons to do this. You can be a monocotyledon, or a monocot, or a dicotyledon, a dicot. So we'll see what they are now. Now, monocots have one cotyledon in the seed, okay? Dicots have two. It's pretty simple. So let's have a quick look. Corn is your very typical monocotyledon. So we can see the structure looks like this. You've got the seed coat, the endosperm fills up there. You've got the single leaf, which is the monocotyledon. Then you've got the sheath structure, so the leaf sheath, which is this covers what we call the first true leaves, which are in here, and then we've got the radicle down here. It's the that's that going on. So the cotyledon is this leaf that sort of covers everything on the outside. They're non-photosynthetic for monocots, whereas a dicot, they're photosynthetic, so they'll pop out of the ground like they do here and start photosynthesizing straight away. Dicotyledon leaves, seeds are actually a bit simpler. So you've got the seed coat, the endosperm, the two cotyledons. Oh, sorry, that's two cotyledons here, and you've got the first leaves pop up. So that above there's your plumule, and down here's your radicle. And we can see it. See, you can see how it shoots out of the seed there. There are a couple other defining features which we'll get into in a little bit. But monocots have parallel veins in their leaves. Dicots have, what are they called? Net-like veins. And we'll see a few of those in a minute. But here we see that's a monocot shoot. That's a dicot shoot. They're actually quite easy to tell the difference between. All right, so we're probably not going to go. So very quickly, we have fibrous roots for a monocot and a long taproot with anchor roots coming off the side for dicots. So that's just a couple of the differences. We'll go over a few more differences in class. All right, so germination. This is basically the process of where it grows from a seed. It is not a seedling process. It is where it first pops out. The seedling first pops out. So it's the process by which the plant grows from a seed. That's germination. The most common example is the sprouting seedling. We've all seen it before, but we've also got a sporling growing from a spore in like a fern or whatever. That's also germination. So here are a couple of the examples of the processes of what it looks like. So you get the radicle pops out. The hypocotyl, which is the bit before the cotyledon, pops up. Epicotyl is the bit after the cotyledon. So you see hypocotyl, cotyledon, and then the first leaves pop up. That's the process. Here with the monocot, we've got the seed pops up, okay, the epicotyl pokes out, the hypocotyl's beneath it, okay, and see the cotyledon is this area in between. All right. So what are the factors? How can we change germination? Basically, seeds are pretty dehydrated. They've only got about 10 or 12, 15% of water in there. So and usually a plant will be 70 to 90% water. So they're fairly dehydrated. So they need to absorb water in before they will germinate. Because otherwise they just can't do it. So water causes the seed to get soft as well. And it allows it to rupture. So it allows it to burst through. So if we go back here, you can see that once you've wet it, the epicotyl or the root, the radical, depends. See, the epicotyl comes out this way, the radical comes out this way. So you can see that the epicotyl or the radical is able to push out because the seed's gotten softer. The seed coat has. Yeah, okay, so oxygen. They basically don't need a lot of oxygen. They don't need a lot of oxygen when they're a seed. Yet they respire a lot. So that aerobic respiration we looked at, which uses oxygen, they need a lot of that when they start to grow. So if your seed is buried too deep and it can't get the oxygen out of the soil, it won't germinate. Alternatively, if you cover it with too much water, it won't be able to get the oxygen out of the little air pockets in the soil, so it won't germinate then either. Effective light varies. Some seeds are unaffected, and you can do this with an experiment to find out. Some seeds are unaffected, while others require huge amounts of light. And temperature. Look, can take place over a huge range of temperatures. 5 degrees to 40 degrees in biology is a massive range, but it's usually between 25 to 30 is ideal. And this gives energy for those reactions to take place. So the respiration and all the stuff which is causing it to move, it won't happen if it's too cold. Now, how can we test this? It's very simple. We put some test tubes in, cotton wool down the bottom, and we'll test this in class. And you put a bunch of seeds in, and here we see we've got here we've got water, oxygen, right temperature, and light, and it grows quite well. Here it's got everything except for light, and you can see that it germinates fine, but once it's above the ground, it's quite weak. Here in the fridge, it's not going to happen. With no oxygen to absorb, it's not going to happen. And with no water, it's not going to happen. So we can test that in class, and we will. Hope that was good. See you then.